My name is Mike King, and I'm a biotechnology analyst and managing director here at H.C. Wainwright. While we're virtual this year, we're confident we are going to be able to provide value to you with over 850 companies presenting at this conference, as well as your interaction through one-on-one meetings. For those of you that don't know us, H.C. Wainwright is a full-service investment bank dedicated to providing corporate finance, strategic advisory, and related services to both public and private companies across multiple sectors and regions. We have a total of 19 publishing senior analysts and over 560 companies covered across all sectors. You may visit our website at hcwco.com for more information. From a logistics standpoint, please make sure to reference your virtual conference online portal that provides your individual links to your meetings and all presentations. Please also join us for corporate presentations and panels that will be available live and streaming beginning on September 13th and running through the 15th. With that said, have a productive and enjoyable day. Now, we'd like to kick off our panel, Engineered Cytokines, the next big wave in immuno-oncology. I have to say, personally, I'm very excited about this panel. We've got a lot of excellent guests who are going to be very interactive. We're going to be talking about this sector for over an hour. And I believe that this is an investable theme in 2022. I've said to a lot of investors out there that we believe 2022 is going to be the year of the engineered cytokine, and that this area will be an investable theme that will benefit not only the companies that participate in this space, but the entire biotech sector. Now that we've got the formalities out of the way, I'm going to go ahead and ask our guests to briefly introduce themselves and give us a quick snapshot of their background and where they fit into this vast space. So we're going to start with our academic participants. We have a long list of questions and topics to discuss. Hopefully we'll get through them all in the time allotted. Let me apologize in advance if we don't. So let me start by introducing asking our academic participants to introduce themselves. Dr. Jamie Spengler, can we start with you?
Absolutely. Hi, everyone. Nice to meet you. I'm Jamie Spengler. I'm an assistant professor of biomedical engineering and chemical and biomolecular engineering at Johns Hopkins University. My background is in protein engineering with particular focus on antibody and cytokine engineering. And I'm really excited to discuss these exciting topics with all of you today.
Thanks, Jamie. Dr. Atkins?
Sure. I'm Michael Atkins. I'm currently the Deputy Director of the Georgetown Lombardi Comprehensive Cancer Center in Washington, D.C. And I'm a medical oncologist with over 30 years of experience with cytokine work. I began this work when I was in kindergarten. And I have been working with IL-2 since the mid-1980s, led the cytokine working group for 20 plus years. And during that time, we've worked on other cytokines such as IL-4, 6, 12, and 18, and have since been trying to advance immunotherapy through checkpoint inhibitors and trying to figure out how to combine checkpoint inhibitors with other agents, including cytokines, to advance the curative potential of immunotherapies.
Great. And just as far as potential conflicts with commercial companies, can you just tell us who maybe some of the companies are that you are, either consultant with or shareholder, et cetera?
I am a shareholder for Werewolf and Pyxis Oncology. I think I've consulted to all of the IL-2 companies. There may be a couple that I haven't, but currently Werewolf, Asher Bio, and a little bit of discussions with Cytokine.
Okay. And Dr. Spengler, if I could, sorry, I forgot to ask you to any disclosures that you might want to make.
Yeah, I'm on the advisory board for Gilead Therapeutics and BioCurie. Yeah.
Okay, great. Dr. Redmond, can we hear you?
Yeah. Hi, I'm Will Redmond. I'm a member at the Earl A. Childs Research Institute at the Providence Cancer Institute here in Portland, Oregon. I have a preclinical research lab, and I'm also the director of our immune monitoring laboratory. I've been involved in preclinical immuno-oncology studies for many years, and about the past 7 years or so, also helping do biomarker work around patients enrolled in immunotherapy trials at our institute. As far as disclosures, I have a sponsored research agreement with Nectar Therapeutics and have worked with them over the years.
Okay, wonderful. And then Dr. Diab, could you give us your background, please, and any disclosures you might want to make?
Sure, I'll start with my disclosures. I don't have any shareholding. I am consultant for many cytokines companies, but I do have research fund from Nectar Therapeutics and Sanofi, as well as I have an advisory board for medical advisory board for Nectar Therapeutics, as well as a scientific advisory board for Memgen company. My background is a tumor immunology, and that's my scientific background. I trained as a tumor immunologist and working on vaccines and cytokines, and later on, when Jim Allison joined Memorial Sloan Kettering many years ago, I did a lot of combination with checkpoint inhibitors and co-stimulatory molecules, as well as adoptive T-cell therapy. But the focus was on a lot on cytokines. Mostly then was the IL-2, IL-7, IL-15. Currently, I'm a medical oncologist and melanoma medical oncologist, associate professor at MD Anderson. My research focus on intratumoral therapy as well as cytokine therapy, mostly IL-2 and IL-15, but also the evil cytokines, that's the name for them, IL-1, IL-4, IL-6, and the focus on how we use these combinations to traffic to the right anti-tumor immune response without exacerbation, the autoimmune response that we're seeing it. That's it.
Wonderful. Thank you so much. Always helps to have the perspective of a clinician with the kind of experience that you have. Okay, turning to our quote unquote commercial participants, let's start with Deborah Cherish, please.
Hi, everybody. I'm Deborah Cherish. It's good to see some familiar faces. I'm a scientist, and during the pandemic, I co-founded a new company called Raise Bio. So I'm obviously a shareholder there. That company is developing novel radiopharmaceuticals. I did spend nearly a decade at Nectar Therapeutics and actually conceived, patented, and developed the Nectar 214 molecule, also known as bempeg-aldesleukin, as well as an IL-15 engineered cytokine called Nectar 255. And by the way, I do believe the modalities of radiation and cytokines could play well together someday. So that's something to look forward to. For clarity, please note I'm not representing either company today here at this panel, or any company for that matter, just here as myself. And I do have, I am also on the scientific advisory board for Xilio Pharmaceuticals and Oncofa in San Diego.
Great. Deborah is of the school of the you do you philosophy.
Yeah.
Glad you could make it. Thank you so much. Let me turn to Randy Isaacs, please.
Certainly. Thank you, Michael. I'm Randy Isaacs. I am the Chief Medical Officer for Werewolf Therapeutics. My background is I'm trained as a hematologist oncologist. My areas were really in biologics, immunotherapy, and cytokines, and I was in kindergarten with Michael. So, we attended together.
Great. Feng?
Thank you, Michael. I'm Feng Tian, President and CEO of Ambrex. My background is chemistry. Ambrex, actually, we have a very unique technology. We can incorporate unnatural amino acid into any protein using E. coli and the mammalian system. And using this platform, right now we're developing a bunch of product lines, including ADC and also IO. In our IO portfolio, we have engineered IO2 that is ready to go to clinic next year. Very happy to be here.
Yeah, and we're really pleased you could join us, Feng. Thanks so much. John Descharlais, please.
Yeah, thanks, Michael. Hi, John Desjardins. I'm the Chief Scientific Officer at Xencor, where I've been since 2001. Xencor has a growing pipeline of immuno-oncology programs, including multiple bispecific checkpoint inhibitors, multiple T-cell engagers, including either engaging CD3 or a preclinical CD28 program. But importantly for today's discussion, we also have multiple cytokine programs moving forward. So we have a long-acting IL-15 FC that's in phase 1 that's partnered with Genentech. We have a phase 1 Treg biased IL-2 FC for autoimmune diseases. And we have a preclinical IL-12 FC program that we hope to file the IND in the middle of next year.
Great. Quite the breadth of exposure here. Fahar?
Hi, hello, Mike. Nice meeting you. Thank you for inviting Medicenna to this presentation today. So want to give you a bit of background on myself. Unlike others, my training has been in biochemical engineering. So I'm more focused on the engineering side of things, but nevertheless have founded Medicenna. I'm the president, CEO, and a director of Medicenna. And what we are doing at Medicenna is really focused on developing and building out our Superkine platform, which are really engineered interleukins. The focus very much on IL-2, IL-4, and IL-13. And these molecules are either agonists, superagonists, antagonists, as well as partial agonists. So really excited with the pipeline that we've built, looking to imminently enroll our very first patient in our IL-2 program, as well as building out a pipeline on bifunctional superkines. We call these assets biscuits. We think the ability to create different flavors of biscuits with different cytokines and antibodies will really set the stage for the company's growth. Thank you.
Terrific. Thanks, Fahar. And yes, as you guys have seen in our list of topics, we definitely want to get to the topic of bifunctional, bispecific, tumor microenvironment activity, et cetera. So we're looking forward to that discussion as well. Anish Suri, can you please kind of introduce yourself?
Sure, Michael, thanks for having us. So my name is Anish Suri. I'm the president and CSO for Q Biopharma, a small publicly traded biotech. Q has sort of taken the perspective from the T cells lens. We've been focused on a very seminal immunological question of signal selectivity, which is how do you ensure that the activation signals are delivered to disease-relevant T cells? So the platform we've developed is called Immunostats. It harnesses the specificity of the T cell receptor to selectively target relevant molecules to tumor-specific T cells. The first series, Q100, is an IL-2 variant on the scaffold. We've taken that into the clinic as a monotherapy. We've reported on early data, including confirmed PR that we've observed already in the dose escalation phase in recurrent metastatic head and neck cancer. But we've dosed this up to 8 mg per kg with no MTD, obviously with the IL-2 variant that's been engineered. But that's to the central thesis of how we've looked at this in context of delivering to those T cells that have a relevant specificity for disease antigen. So we're very excited to be here and very much look forward to participating. And again, thank you for the opportunity.
Yeah, absolutely. Glad you guys could make it. Very interesting story and a unique take on guided IL-2 therapy. And then finally, last but not least, Jennifer Michelson, can you say hello to us, please?
Hi, Jennifer Michelson. I'm the Chief Development Officer for Biologics at Cullinan Oncology. We're a small public biotech in Cambridge, Massachusetts. And I've been in the industry for about 20 years or so, with the last 10 years really focused in cancer immunotherapy, in particular working with Third Rock Ventures to help to launch a couple of new companies, including Jounce Therapeutics, which was really one of the first IO-focused companies where I had the opportunity to work with Jim Allison and to lead the flagship GTX 2011 program. And I joined Cullinan Oncology about 3.5 years ago. And here I oversee the biologics portfolio. We have a very broad IO pipeline, including multiple T-cell engagers, various multi-specific therapeutics. And specifically, of relevance to today is a very exciting cytokine platform that we have. It's our AMBER technology, which is designed for tumor retention of cytokines. And in particular, we're developing a dual IL-2, IL-12 tumor retention based therapeutic that we're excited to be able to talk a little more about today.
Great, Jennifer. And just for a point of clarity, because I don't know the Cullinan story super well, was that an in-licensed molecule, or is that something that does germane to the Cullinan platform?
So the AMBER technology in particular is, we licensed the technology from MIT, but the actual molecule that we're developing is sort of wholly owned by Cullinan in terms of the intellectual property. So,
right.
Yeah.
Okay. All right. Super. Sorry for the folks out there who are watching. Apologies for a little bit of a long introduction, but I think you'll agree that we have a vast array of experience here and a wide swath of exposure to the cytokine world, sort of broadly writ. So with an emphasis on IL-2, but clearly there's a whole lot more going on with things like IL-12, 15, et cetera. So let me, everybody on this call pretty much has heard my spiel, and I apologize for making you go through this again, but I think to set the stage, I'd like to relate the story that, that prompted my thought about creating a panel such as this. And it was really the flurry of financings that have recently taken place in the engineered cytokine world. That was kind of a wake-up call for me. As the listeners may know, in full disclosure, H.C. Wainwright was a participant in the initial public offering of the Werewolf IPO. And it was interesting from my point of view because I had to go back to these publications from the 80s and 90s and reread stuff that I had learned about from the early, the sort of the dawn of my career. I was probably in pre-K at the time. But in June, we saw two private development stage companies obtain significant amounts of venture capital to fund the development of their respective pipelines of engineered cytokines. These were companies that you've heard one name, Cytokine, and the other was a company called Bright Peak. And then recently, September 1st, we saw yet another $108 million round closing for Asher. Deborah mentioned Zilio, and they haven't funded recently, but they also were funded some time ago with quite a bit of capital. So I found it fascinating that there was this, this intersection of investment in this space. And the other observation that I made was that RA Capital, which many of you know, has made 5 bets on this space. And those guys have a pretty strong track record of accomplishment. So I'm going to throw this open to the panel. I think anybody can jump in. I think we should maybe start early in the days for the folks that were involved with BEMPEG, maybe chime in first so we can get some perspective of history and then build upon that. But why is all this happening now? Was there some kind of a fundamental breakthrough that took place recently? Or is this kind of, a cynic might say, well, this is just the typical behavior that one sees in the venture community. It's someone sells a company for a lot of money, or someone does a big deal with a major pharma company, and I want a piece of that, and I'm going to form my own company. So maybe we can talk a little bit about the fundamentals that are going on. Is it bigger than all of this, or what? So maybe, Deborah, since I think you've got the benefit of perspective, maybe I can start with you. Kind of how did you initiate, conceive of BEMPEG, and in your perspective, what are these other modalities coming through say to you?
Sure. Thank you, Michael, for this opportunity. I actually would call this more like a second wave of cytokines, because we did have a first wave in the 1990s, and there were, in fact, a number of cytokines that were approved, IL-2, interferon, alpha, beta, and gamma, GM-CSF.
Alpha interferon,
yeah. Yeah. So in my opinion, the key difference is I think that we fundamentally just understand the immunology better. So we have a better understanding of, which cytokines are addressing which cell types and what are the effects on those cells and their signaling. So that then helps us engineer cytokines to be better drugs because we're no longer wedded to those endogenous cytokines from the 1990s. We can tinker with them. So, what we wanted to address, for example, with things like, well, IV dosing every 8 hours doesn't sound like a very good drug, but IV dosing every 3 weeks, well, that starts to sound like more like a real drug, or compared to, say, a continuous infusion in an ICU bed. So, the first thing, that we thought of at Nectar was we really wanted to take high-dose IL-2 and make it be outpatient, first and foremost. And that's how we initially engineered Nectar 214. So that was a major design goal for Bempeg back in 2010, which is almost feeling like kindergarten now. But at the time, that outpatient dosing was, in fact, a big deal. So I think, there's a constellation of things, but I would think that a deeper understanding of the immunology in order to better design the drugs, in order so that physicians feel more comfortable dosing it, especially dosing immune modulators. So I think those things I think have come together in the last decade, to expand the role of cytokines in the immunology space.
Okay, maybe I could ask Dr. Atkins, Dr. Dion to contribute here. One can, I think the dosing of cytokines has always been a bit of a, art more than a science. And, what do you feel that there was adequate time spent trying to figure out dosing? Some investigators still are using high dose IL-2 with, and hospitalization versus others where, it's more of a metronomic or low dose approach. Have we basically wrung all the juice out of the lemon, so to speak, of the native cytokines?
Maybe I can talk about that because I spent about 20 years of my career trying to address exactly that question. And I think we explored all the different ways in which we could give IL-2. First IL-2 with lac cells, and then we showed that the lac cells, which were the peripherally activated T and NK cells added to the high-dose IL-2, didn't add anything. Then we looked at continuous infusion IL-2, and we found that although we could increase the T cell numbers in the periphery and the rebound lymphocytosis, which could help potentially in generating more lac cells, we didn't see the same degree of efficacy, and we didn't see any reduction in toxicity despite using a lot lower doses. We then looked at combining IL-2, high-dose IL-2 with interferon in subcutaneous regimens, which could produce responses, but didn't produce the same degree of complete responses, which are cures that we saw with high-dose bolus IL-2. We looked at combining with chemotherapy, which all the way to a phase 3 trial in melanoma in the cooperative groups, which even though we had promising phase 2 data showing increased response rates, increase in the median progression-free survival, we saw no benefit in overall survival compared to chemotherapy alone, suggesting that chemotherapy was inhibiting the activity of immune therapy. And then we looked at ways to dissociate the toxicity of high-dose IL-2 from its anti-tumor effects, combining it with steroids, which allowed us to give 3 times the amount of IL-2 without toxicity, but also without efficacy. We looked at selectively blocking IL-1 and TNF, which did not block the toxicity, but also maintain the efficacy. And then failing that, we looked at trying to identify who are the patients who respond to IL-2. And we identified a group of patients who had inflamed tumors, including PD-L1 expression as potentially predictive markers for IL-2, all of which I think set the stage for checkpoint inhibitor therapy, but left us with a drug that could only be administered in order to produce cures the way we were doing it. with to a small percentage of patients with melanoma and kidney cancer who could tolerate it at institutions who were willing to build teams to be able to deal with the side effects.
And how great were those benefits in terms of added progression-free survival or overall survival?
Well, we never looked at progression-free survival because it didn't budge. Overall survival didn't budge, but somewhere between 10 and 15% of patients were cured of their disease.
Okay.
And so that was proof of principle that the immune system, if properly activated in the right tumor microenvironment, could produce cures. And although few people actually thought that was a useful way of treating cancers at the time, it did keep the field alive until we were able to identify better ways of activating the immune system.
Okay. And Dr. Diab, you have a vast amount of experience. Any thoughts of what you could add to Dr. Atkins' comments? Oh, sorry, Dr. Diab, we seem to have lost your audio again. There we go.
Big to add to Dr. Atkins' comments. He definitely pioneered the field, and his experiences is really what pushed the second wave, I think so, for cytokines. My experiences is although I worked with overlap with some people who did some experiments that Dr. Atkins described with biochemotherapy and comparison and combination with interferon alpha. But my experiences comes from the two extremes of high dose IL-2 and low dose IL-2, and that described the complexity of IL-2. And definitely, I started my immunology career with the introduction of T regulatory cells by Shimon Sakaguchi, and we start learning about that and the important role of IL-2 there. And my clinical experiences, both with high dose IL-2 and low dose IL-2, really describe where IL-2 can really be a very beneficial cytokine or very suppressive cytokines, if we want to use that in the setting of tumor immunology. And I think that's where we focused our science, and that so that the experiences is right now that low dose IL-2 is certainly an important molecule for immune suppressions, and high dose IL-2 could, in certain populations, could be very important cytokines and pivotal and non-overlapping with anything, and its effect cannot be achieved by any other agents, maybe some other cytokines, but I do believe that even with the beta gamma, that the IL-2 firepower is exceeding a lot of the other cytokines. And our focus is really on our experiences that preclinically is when you add IL-2 to any combination in the preclinical model, it's better. It's always better when you combine with the checkpoint inhibitors, with vaccine, with adoptive T cell therapy, with co-stimulatory molecules, with innate activating agents, TLRs or CD40s. Whenever you add IL-2 in the preclinical models, the outcome is better. One of my very well-respected colleagues right now, he's in Nectar Therapeutics, but for many years he led that field as well, William Overwick. And he had a lot of experiences as well, like me, with combining agents, and he has those experiments and showed me the data that when you add IL-2, you have better outcomes. So the need for IL-2 to be translated into the clinic based on this and based on what Dr. Atkins just described has been eminent, especially with the new wave. The new wave is really built also in the opening of the immuno-oncology world with the checkpoint inhibitors. So it was obvious that IL-2 is one of the things that we want to reinvestigate. And personally, that's what led me reach out to Deborah and Nectar Therapeutics in 2013 to try to collaborate with their agents that combine those worlds of IL-2, minimizing the suppression and maximizing the efficacy with relatively good toxicity and practical administration to the patients.
So if I don't want to put words in your mouth, Dr. Diab, but if I synthesize what you and Dr. Atkins have said, it sounds to me like IL-2 is going to be, it's like a side dish that goes with everything. It does have its own intrinsic activity, single agent activity, but its real power is going to be in combination therapy. Is that a fair statement?
The Americans call it the bacon of food, so you can add it like bacon with everything.
Right.
And the Middle East call it olive oil of things. You can add it.
All right, good.
Michael, can I add one comment?
Of course.
I think, to the question of why now, I think a huge aspect of that is the tremendous success of the checkpoint inhibitors, basically reopening the field of immuno-oncology. And then a bunch of companies like, Xencor, Roche, whoever, wondering, well, how do we build on that activity, right? What's next? And it doesn't take you very long to zero in on, okay, let's work with cytokines. I mean, there obviously are other approaches as well, other checkpoints, sure. But I think that's a huge part of the watershed now.
Well, maybe just to, John, because I know you guys have other IO assets in your portfolio as well. And I'm coming from a very, obviously a very Wall Street focused perspective. And it just seems to me that the street, I guess was sort of anticipating the next wave after the PD-1/L1 phenomenon to things like TIM3, LAG3, OX40, CD4, more checkpoints and co-stims. And here were, cytokines were sort of, standing right there. And people were not overlooking them, maybe not ignoring them, but overlooking them because of the, the track record of IL-2, the talks that had been seen. But it seems like everybody was desirous of single agent activity, and you already had that with with cytokines. So anyway, it's just kind of interesting how the spotlight went from second generation checkpoints over to cytokines. Not saying it's a bad thing. I think it's a great thing. And I think it's ultimately, and I think we all agree that the real beneficiaries are going to be the patients. But maybe one quick question before we, go ahead,
Fahar here. I think based on what Adi mentioned about the olive oil story or the bacon here, really it seems like IL-2 will be a prerequisite for various other therapies. So the opportunity is obviously quite great, especially with the advances that have taken place in engineering these cytokines or creating safer, more effective versions. And I think one of the watershed moments was clearly the very large BMS Nectar transaction that took place, 4 years ago. And that's basically, I think, from an investor perspective, provided that additional external validation or a big pharma validation that the IL-2 is the space to go, despite the fact that there was little sign of monotherapy or single agent activity. So I think it created that momentum amongst all the others who are working on the engineering side to sort of create a better version of IL-2 and hopefully have an opportunity to address so many different cancers that are obviously not being fully leveraged by the checkpoints out there. So
well, I agree with you, Fahar. It's just, again, I think correct me if I'm wrong, I think the Bristol Nectar deal happened in like 2018, was it? Deborah, maybe you?
2018, yeah.
I think they closed in early 2019 is when it actually closed, Yeah.
Okay. So maybe it just took that long a time for, the engineering to catch up. But just before we kind of, I don't want to leave a single agent IL-2 just yet, but I'm just curious if our clinicians or anybody thinks that have we maxed out single agent IL-2 activity? Is there anything that we can do with any of these novel constructs to enhance this, single digit or low double digit cure rate that we see with the native molecule, based on some of these, again, modified versions?
If I could chime in, sort of from the Cullinan perspective, I agree with what everybody said in terms of really trying to build upon, those early findings with the known activity, the clinical efficacy, which, you can't ignore from the early studies with IL-2. And so, part of it is just going back to getting a better understanding of biology and understanding or just thinking about the biology. And we think about cytokines, they're naturally occurring proteins, of course, that typically are functional in a localized tumor microenvironment or localized environment in general. And so the notion of systemic administration is perhaps something that, not surprisingly, may not be the most efficient or effective or safe way to actually administer the cytokines. And so there are obviously a lot of different ways to skin that cat, but one of them is to consider intratumoral delivery, and that's one area where certainly a number of companies are focused with cytokines as well as with other agents. But there's certainly, intratumoral delivery on its own actually has a limitation that, of course, the cytokines can just leak out. And so it's not necessarily a panacea in terms of solving the toxicity challenge. And so, the approach that we take at Cullinan is engineering the native cytokines. In our case, we're working with IL-2 as well as IL-12 in a single therapeutic. But we are actually using collagen binding domains as a means to retain the cytokines within the tumor microenvironment. And so you get local delivery as well as retention of the cytokines. And so you can have actually localized high concentration cytokines, which is essentially how cytokines are normally functional. And that way sort of maximizing the efficacy and reducing the systemic toxicities that would otherwise be happening with systemic administration or even intratumoral administration without local retention. And so we have built this AMBER platform for our dual cytokine delivery that enables, essentially delivery of, in our case, two potent cytokines on a single molecule with a collagen binding domain, IL-2 and IL-12, for, what we're seeing, at least preclinically, very potent anti-tumor activity, and with the potential to actually work in a broad variety of solid tumors.
Right. Dr. Diab, any thoughts on the local? I mean, how straightforward is it to do? There are other approaches for local administration, direct administration to tumors like TLR agonists and other things. Do those, typically, how did the medical community view those? How do patients react to those kind of approaches?
The intratumoral field is very interesting approach, and certainly one of the approaches that scientifically and immunologically make a lot of sense. In a practical situations right now, the intratumoral field is in a major skeptic mode after 2 years of 2 phase 3 trials. One is the master key of collaboration between Merck and Amgen of pembrolizumab plus TVEC versus pembrolizumab alone. And certainly there was interesting phase 1-2 published by Tony Ribas and very interesting neoadjuvant approach, but the phase 3 failed. And another trial that I was personally involved, which is the intratumoral tilsitulimab TLR9 agonist plus ipilimumab versus ipilimumab alone in the refractory. And that there was a press release from IDERA that the trial failed to achieve its primary endpoint. Now, again, between the concept and its implication, there's million barriers, and to optimally inject intratumoral without just deviating the conversation to that, there are barriers. The concept is extremely important. I think using the tumor as vaccine site is very important. There is success preclinically. Just bringing it from drug development and make it phase 3 trials, there's a lot of major barriers. I think they are going to be studied well, and hopefully we can overcome them. For that, the SITC actually there is a session this coming SITC to discuss intratumoral therapies at a crossroads. And I encourage you all to attend it. It's going to be chaired by myself and Nina Bardawaj, and there will be a lot of experts in intratumoral. So they will discuss these approaches, the benefits and disadvantages and the barriers. Just to have one more comment on that single agents activity, that's what you wanted to see of IL-2. I think the drug development and the way it is right now in the clinical trials, you have to appreciate the differences between the patients that been tested, that Dr. Atkins initially reported them as the first report on the importance and the activity of IL-2 versus the patients that are being tested right now with these modified IL-2 agents. And there's a lot of these patients, as Dr. Atkins mentioned, he identified patients who have inflamed tumor microenvironment. Those patients, there's a big overlap with them, with those patients who benefit from initial checkpoint therapy, whether it's PD-1 or CTLA-4 or combination. So So you squeeze them out, and then you end up with refractory populations. That is not necessarily the population is going to benefit from the IL-2 pathway. So the refractory population is sometimes squeezing out the single agents. And we know right now that the entry to immunotherapy has to come through checkpoint inhibition. That's where is the major things. Doesn't matter how much you stimulate, you probably need the checkpoint inhibition. IL-2 has evidence that you don't need that. That's what Dr. Acton's initial reports showing with the initial response. But at the moment right now, the entry to the field is through checkpoint inhibitors. So the drug development is not the same patient populations. We're looking for single agents activity, unless we take it into the neoadjuvant setting or the treatment naive settings, which that might be something we should look into it.
Oh, well, it's interesting that you say that. I just wonder, one of the things that, and I may be misinterpreting, the science here, so forgive me, but I noticed, one of the aspects of BEMPEG that I found interesting way back when they first reported data, I believe it was in the melanoma setting, was that they actually had activity in PD-L1 0, PD-L1 low patients where, single agent activity wouldn't have been expected. So is that, I just don't remember how many prior therapies those patients had, or et cetera. So maybe you can illuminate that for us.
I think that what you're talking about is still in the combination with nivolumab. That's where the
Right.
data that is published, that in combination with nivolumab, there has been responses in patients who are immunologically poor, who don't have interferon gamma gene signature, or don't have high expression of PD-L1. And that's, you're definitely going to see and ask your question, how is this maybe contribution of the PEMPEG? And certainly that's something we think and we believe it is, but this was single-arm trial. You still see some activity of nivolumab or pembrolizumab in PD-L1 negative and interferon gamma negative. These are correlative markers, but by all means not absolute predictive markers, where if you don't have them, you're not going to see activity of anti-PD-1 or anti-PD-L1. So there is hint that PEMPEG can overcome for sure, and the biopsies is indicating that there is activities of PEMPEG alone in these situations, but you have to do randomized trial to demonstrate the superiority of a single agent in that scenario.
Yeah. All right. Well, I want to get some of the folks that haven't contributed yet, excuse me, into the discussion. And I know that, like, say for you guys, Randy, at Werewolf, you've done and again, we have to take the animal models with a grain of salt, but, you've shown your 124 against native IL-2 in the MC38 and the CT26 models, and they look wonderful. I'd love to hear from Feng and maybe, Fahar, Anish, you guys please feel free to contribute. But, single agent activity seems, with your construct, Randy, to produce superior results. And, maybe you can comment about how confident you guys would be that you're going to get to something close to that in human studies.
Yeah, I
So,
can.
go ahead.
Randy, go ahead.
No, go ahead. You go first.
Thank you. Yeah, well, we can, talk about this, from, Ambrx's point of view. We, in essence, a technology company. It's just like, HAR, we are coming into the door from an engineering, point of view. And what we're trying to do is actually we are using our technology to integrate many aspects of drug development, including small molecule, large molecule, polymer, you name it. Okay, so it's more of engineering coming in. So this really leads to another question. It's the first question you asked, Michael, is why now, right? Why now is what I see is, first of all, IL-2 has been probably the best studied cytokine so far. Thank you, Dr. Atkin's contribution. Number two is nowadays a lot of technology is available, especially when you look into using unnatural amino acid. What's the advantage of that is really give you another dimension of universe for engineering that is not available before. Okay, and also give you a tool to precisely engineer in a chemical point or chemical way. Okay, what I mean by that is we can engineer by the precision of atom. Okay, and we can add in switch, take away switch, okay, add in function, take away function. It's a completely new game. So right now, I think the tool is available. And also, I question, why we have to pursue single agent? Why? Okay, because our goal, I believe, is to create a cure for the patient, right? So single agent, double agent, I think combination is always the best. And
I think there's no doubt. But I think most KOLs that I've spoken to over the years have felt that most and any IO agent that lacks single agent activity is not going to be additive when combined. So even if it's a hint, but on this at the same by the same thought process, there are plenty of patients out there that have blown through their checkpoint inhibitors, and it would be helpful to have something that would work in that setting, unless maybe Dr. Diab would disagree because maybe their immune systems are completely fried and wouldn't respond. It would be I would think it would be helpful to have something that's active as a single agent.
Yeah, and I think just to add to that, clearly there is also the ability to sort of create these bifunctional cytokines where you have both activities within the same molecule. An example is Roche's molecule where they have a cis fusion with an anti-PD-1. This is something that we're also working on, or having bifunctional, or I think Jennifer mentioned the IL-2, IL-12 combined within the same molecule itself. So those approaches obviously will further improve sort of the therapeutic profile of a drug, where you have a bifunctional or trifunctional activity within the same molecule, just as we have seen the adoption of bispecific antibodies. And I think one other thing that you brought forward about localization was, how do you sort of administer the drug systemically, which is obviously the most convenient way of administering the drug, but at the same time allow it to accumulate at the tumor or at the draining lymph nodes where the immune system is more active. And I think one of the things we've done is create fusions with albumin, and then that we know tends to accumulate in the tumor site very effectively. So I think those, subtle but interesting changes that you can make on the design of a molecule is there potentially to further improve the outcomes from a safety efficacy perspective, notwithstanding the fact that there is a possibility, even with these bifunctional superkines or cytokines, to combine with other therapies, including radiation and so on and so forth. So I think there is, and I think Randy probably has a sort of a different take on it as to how they are able to sort of activate the molecule right at the tumor site itself.
Yeah, I do want to. I do want to, Fahar. I appreciate you bringing up the bifunctional molecule. I do want to get to that, but I want to try to stay on track with single agent first, and we can then go to bifunctional. But it's definitely something that is worth discussing. So, Randy, if you could just comment on the werewolf approach.
Sure. So werewolf therapeutics originated from an interest in the pro-inflammatory cytokine treatments for cancer, looking at technologies where you could use a modular approach in order to be able to target the tumor microenvironment, yet successfully mask the cytokine in the periphery and allow for delivery of a fully potent cytokine to the tumor, which would allow for complete biology to be seen. I think it's important to emphasize that our goal in designing these optimal cytokine therapies was to solve for several pharmaceutical property issues, to get around the idea that you need intratumoral injection. And these are the basis for the challenges with this class of agents. So our comprehensive approach, our predator platform, which yields what we call our indokine molecules, the first two being WTX-124, IL-2, and WTX-330, IL-12, were designed specifically to be turned off in circulation and normal tissues, and at the same time to be delivered into the tumor microenvironment and to allow for delivery of the cleavage and delivery of the fully potent cytokine. I think that lends itself to the fact that we fully expect to see monotherapy activity, at least as good, if not better, than the native cytokines, because they were limited.
Right.
They were limited in the number of patients who could be treated. They were limited in how much exposure one could achieve because of the poor pharmaceutical properties. So combinations either with other IO agents or other standard of care treatments will continue to be the mainstay of oncology therapy. But given the number of IO combination trials that are being performed, we need to be very careful about how best to utilize resources and provide most promising studies for patients. There is a continued need for compounds which have monotherapy activity, especially in the relapsed refractory setting. And as I said, we expect to see monotherapy clinical activity with our Cytokines, something that hasn't been seen with some of the competitors in the field, especially in patients who have progressed on or after checkpoint inhibitor therapy.
Randy, did Werewolf at any point consider bifunctional molecules?
Not in the initial, not in the initial foray.
Okay. I haven't gone. I see Anish. You read my mind. You haven't contributed yet, but it seems like a good opportunity for you to check in.
It's a fascinating discussion, I think, from what Dr. Atkins mentioned from the initial get-go. But there is an immunological elephant in the room as you think about systemic cytokines, which is the frequency of T cells reactive to the antigens that a host has are very, very minor. So the struggle is, how do you get these to those T cells? Localization is an area, if you make the assumption that the tumor is harboring these specificities. If not, you have to induce them. So that's why when we designed our platform, we went with an approach where you could have the signal 1, which is a stabilized peptide HLA, as a dimeric stable construct that engages specific TCRs.
Right.
And an IL-2 with it that uniquely would act upon only those T cells whose TCR is engaged. We've now taken it into the clinic. We see in head and neck cancer patients, to what all of you have said, these are late stage who have failed pembrolizumab, who have failed nivo, who failed first and second line therapies, where we can see T cell responses when biopsies, we're seeing infiltrates, and in dose escalation, we've seen frank anti-tumor efficacy. So to your point of single agent, I think context and specificity and selectivity could get us there. And I think it's all building on the studies of the giants that have been mentioned here to get to that level.
Right. Well, you guys were insightful in, targeting the EBV antigens. But does that, does that mean HPV, sorry. But does that mean that one has to have an identifiable tumor antigen in order to, to get that kind of reaction?
We've done it two ways, Michael. We've gone after primary determinant, well-known drivers, HPV, viral proto-oncogenes, Wilms tumor 1, KRAS, G12 valine, and we've released that data. But we've got a novel class of bispecifics where we are developing, where we can harness the viral repertoire that all of us harbor, the memory repertoire, your CMV, your flu, I would argue even the SARS-CoV-2 at this point, with these immunostats that can coat a cancer cell. So the cancer cell appears like a virally infected cell. We just released some of that data at the New York Academy of Sciences earlier this year. And that's a novel bispecific because that's a tailored combustion of your protective viral repertoire that a host has harbored for decades to then recognize the cancer and destroy it, as opposed to the carpet bombing systemic with broad T cell engagers. And that again came from this thinking on how do you tailor the immune response to a subset that allows you to avoid the systemic toxicities while sort of hopefully gaining on the efficacy. So we call them ready stats, RDI stats, redirected immunostats, but that is a sort of the next generation. Get after that. The single agents are these primary viral proto-oncogenes, Wilms tumor, KRAS, where we have good evidence with a lot of these targets that signal specificities can provide anti-tumor efficacy, of course.
Okay. Go ahead, Deborah.
Yeah, if I could just add something maybe slightly controversial, talking about single agent activity. I'm not sure with immune modulators, that immunologically it's the right way to go, because in the end, what are we looking for? We're looking for a killer T cell, right? An activated T cell is in the end the drug. that's going to kill a tumor.
Right.
So when you have an activated T cell, that thing is going to start secreting interferon gamma. And it seems like, from what our observations are to date, that when you have that interferon gamma response, the last line of defense for the tumor is to do something, upregulate PD-L1 or a checkpoint, right? So then, even in the face of a very strong immunological response, we may still be faced with that checkpoint, and it may be difficult to get around that. So, I do think that in the end, some kind of combination with a checkpoint just makes sense immunologically, even though we might want to see or like to have single agent activity. I think if you're going to have a good immune response, you might need that checkpoint in the end.
I would agree with you, Deborah. I think that's a very good point. I just also go back to some of the earlier studies where PD-1 signaling is reversible by IL-2, et cetera, et cetera. So again, in context, I think if they were there at the right appropriate cell types and in the microenvironment, I think you're absolutely right. And that again goes back to the point of perhaps even the IL-2 pharmacology being focused in these contexts. Entirely true.
So Michael, I think you're hearing, two different stories here. You've got, I actually agree with Dr. Diab said. It's a challenging hypothesis to test whether or not these new engineered cytokines are going to have single agent activity, because you're in checkpoint refractory patient populations. And then I think the other side of the story is we all hope we're going to get single agent activity. Of course, investors love it. That's probably what you're hearing, right? It's, of course we want to see that. And everyone's trying to improve the therapeutic index compared to the native cytokines, right? So, the Q approach is like that. The Nectar approach is like that. The Xencor approach, we discovered a whole different way to tackle the problem. We found out that by reducing the potency dramatically, like taking an IL-15, if we take a wild type IL-15 on top of an FC and put that into primates, it disappears.
And is
within about a day.
Is that potency less than the native IL-15?
That one is about the same. And
okay.
it despite the fact this thing has an FC domain that should promote recycling, it disappeared. So then we realized, wait a minute, let's reduce the potency. And what we found is we can reduce the potency about a 100-fold. And a lot of people struggle with this concept, but what happens is the molecule sticks around so much longer.
Right.
Because when these things talk to the receptors and signal, they actually get cleared. And so the game we're playing is finding that sweet spot between potency and exposure. And, sure enough, if you reduce the potency dramatically, you can actually get far superior exposure.
And you get less tachyphylaxis.
You get less toxicity. Yeah. You get less toxicity. And you get dramatically superior pharmacodynamics.
John's absolutely right. I think that is a very key concept of playing with signal amplitude by dialing out the affinity, something we've played with ourselves. But I think that's a very good point that John raises here in terms of understanding that better. And that this, frankly, has come from a lot of good protein engineering and understanding structure-receptor interaction interfaces.
And I'm curious, Deborah's perspective, because, NACTR 214 has a lot of different states. And so, which one of those states, because they're all going to last longer, right, than the native, than the bare, naked IL-2 once all the pegs fall off.
Yeah, and I fully admit, we took a much more cruder approach than for example, Werewolf. But we did take a pro-drug approach where we didn't want to have an instant immune activation as soon as the drug is injected. So the immune cascade kind of unfolds as the molecule unfolds, if you will. So so there are many different states, each of increasing activity. It's a pretty novel design, I suppose, in that respect. But eventually it does get to the most potent, which is the single peg IL-2. But to your point, these things do coexist together, and but the immediate exposure is not there. As if when you're giving high dose IL-2, you have an instant immune activation systemically, and we believe that that is at least a contributor to the toxicities. But having that IL-2 activity unfold over time, sort of in the way like a T cell might, it's happening over a time span. we think could minimize that, and in fact has been shown in the phase 1, by being able to dose it outpatient. That sort of made the point about the safety component.
Is there also some kind of feedback inhibition, so to speak, from the immune response standpoint? In other words, you overstimulate too quickly, you get a much greater response to that in terms of downregulation.
There is
that's something we're very intrigued by.
Yeah.
Right.
Yeah,
if you go, the way I think of it is like you make the best ribs when you cook them low and slow, and that's how we make our cytokines.
Right.
Well, our experience has been quite different with respect to, the talk about, really promising pharmacodynamic behavior of, for instance, our IL-2 agonist, which is really directed towards the beta gamma signaling process. And what is something that we found quite interesting was that with these agonist molecules, you sort of hit the T cells, stimulate them, and we see a very long, prolonged effect that lasts about 2 weeks or so before the population diminishes. And I think there is, ability to maintain this high level of proliferation of T cells and selectively making sure that not only that, what we are able to see is a very robust memory T cell response. And I think being able to simultaneously stimulate not only your CD8 T cells, but also your NK cell population, which is critical for immune surveillance as well, and future tumors coming back up again. The other thing is also you want to build up a population of your naive CD8 T cells, because we know that the tumors will have a distribution of different tumor antigens over a period of time. And with proper antigen presentation, you want to make sure that you have an adequate population of your naive T cells that can be trained so that any changes that occur to the tumor itself, they can be managed by these trained new T cells as well. So it's sort of it's a very complex. It's not as simple as we all think, but I think it's
great points, Fahar. It's about the naive T cells and definitely the NK cells. I know that Werewolf has shown, the action on NK cells as well. I want to just turn back really quickly to Deborah because I want to get Dr. Spengler involved here, which is and ask you about you had a long history with Nectar and Bempeg, but what was it you hope would be achieved by the Xilio portfolio or their assets that you weren't capable of doing it with Bempeg or with Nectar?
Yeah, and again, I'm not representing Xilio. I'm on their SAB, but it's a more targeted approach. Where it's an approach that would seek to unmask the IL-2 activity in the tumor microenvironment. Whereas at Nectar, we let that unmasking happen kind of, organically, when it wasn't targeted. Yeah, it wasn't targeted specifically to the tumor.
Okay. And Jamie, I know you've been involved with Cytokine to some, not directly, but you're an advisor to Cytokine, correct?
No, I'm not.
Or is it Xilio?
I did my postdoc in CRISPR.
Oh, that's right.
One of the co-founders. So, I'm aware, of the work. But actually, what I wanted to bring up, and the Cytokine actually is very relevant to this, as well as some of the things that Fahar was saying, is thinking about single agent, capabilities, and really the question of sort of nature versus just completely blank slate, right? Starting from scratch. And the ideas like Neoleukin and other companies, that are thinking about computational design or
other we wanted to have them. Unfortunately, they were conflicted, but we were going to.
Yeah, no, actually, I worked on two, in my postdoctoral work and continue collaborating with the Baker Lab as well. So I'd say I think there's a lot of potential in these sort of newly designed formats of IL-2 and other cytokines potentially to really sort of flip the script, I think, in a lot of ways. And the reason that I say that is because, if you start from IL-2 as your template and you make changes to it, there's always kind of that muscle memory of, the way that the energetics and the interactions, of the molecule are going to have residual sort of effects. And if you actually start from kind of a blank slate and create a molecule that recapitulates, sort of the binding functions and the essential pieces that you want, but actually completely erase all memory of other aspects of the cytokine, you can fundamentally change the nature of the cytokine. And I think there is some potential to, go beyond what one would normally see with just a traditional single agent, because it's really fundamentally a different molecule.
Interesting.
Terrific. Will, I want to get you involved here. You've been sitting there quietly, just kind of drinking this all in. I don't know if there's some points that you'd like to get across before we move on.
Yeah, I guess the one thing that jumped out at me, I was thinking about the translational aspect of this. And, one thing that we're a center that does a lot of high-dose IL-2 therapy still, as some of you know. And so one of the things that I think I haven't seen really in the current data from a lot of these companies is any kind of head-to-head comparison. The new IL-2 agonists, these engineered agonists, are great because you have the selectivity for the signaling that you can avoid and reduce the toxicity, which would be wonderful for our patients. We've seen that with Bempeg and other drugs in this space. But I think that also gives you an opportunity to increase the dosing. I know Deb mentioned that they specifically went for an outpatient therapy, and there's many reasons to do that, right? For access to the drug and things like that. But I think, if we could ramp up the dosing with some of these engineered IL-2 agonists and avoid and have that reduced toxicity, I absolutely think you would see similar or better single agent activity. I know there's many business development reasons why that may or may not happen in the development of these things in the phase 1, phase 2 trials. But I think that's something to see, that head-to-head high-dose IL-2 versus some of these engineered agonists. If you could get, even if it was a 15-20% response rate, but a reduced toxicity, I think for our patients would be amazing, right? So there's this huge unmet need for these things. And so I would love to see those kinds of trials. This is my community out there and the companies to do these kinds of studies and see what we can find.
Okay. We're gonna need to move on. I have one other quick question, and then I'd like to move on to some of the other cytokines because I crash out in about 15 minutes for the next set of talks. So I do want to just get everybody's perspective, or whoever wants to contribute. We've got the Pivot 02 studies coming sometime between now and the end of the year. So maybe think SITC is the forum for that. Let's just assume, will the outcome, whether good or bad, detract? It may have an effect on investor psychology, but will it have any effect, good or bad, on kind of the psychology of the industry? Will it prompt people to rethink what they're doing, or will people continue to plow ahead and say, well, that was specific to that study or to that molecule, and continue forward? So whoever wants to jump in first on that is welcome to. Otherwise, I'll pick someone.
Yeah, so I'll volunteer.
Go ahead, Fahar.
So I will start, and really, to be honest, not necessarily whether the data is, if the data is negative or positive for that matter. I think the understanding is there. I think the investment community knows what the potential is of IL-2 in general. I think there is an understanding that clearly the efforts that are being made by all of us that we are here today is to really create a better version of IL-2. Did Bempeg address all the issues? I think that's remains to be seen. But clearly, the key aspects are you want to have outpatient administration of the drug. I think that's being accomplished by everyone that we've been talking to today. Are we creating a better, more efficacious molecule? I think that's the attempt. And are we trying to create a molecule that's also safer as well? And I think by and large, the efforts by others are not duplications of what Nektar has done, but quite dramatically different from that perspective. So I think that shift, I think the excitement around the space is going to build. I believe that every attempt to incrementally improve the IL-2 is going to be very beneficial to the patient population that we need to address. And I think the key thing here is also, how do you post-treatment, do you see any changes that are occurring in gene expression in the tumor microenvironment. I think that needs to be monitored, and I think work that Will is sort of trying to do in immune monitoring studies, I think that's important to see if are we creating the tumor and making the tumor microenvironment more inflamed. And we have, for instance, seen with our IL-2 work done by independent labs to show that the gene expression profile is quite different post-treatment with our drug as compared to your native Proleukin. So I think there's a lot more to be learned, and I think there's a long way to go. And I would say that there is obviously room for a lot of us to be in this space. And it sort of complements the fact that IL-2 of some sort will be prerequisite going forward.
Yeah, and I would argue, to Dr. Diab's point earlier about single agent studies are obviously less predictable than the randomized study. It would seem to me the randomized phase 3 melanoma study that's due in the first half of 22 is probably more relevant. But, we do have an investor world that tends to be relatively short-term focused. Anybody else want to address that at all?
Look, I would just echo again, Michael, the comments made by William earlier on, which is having a safer version that likely could be more selective from a patient's lens could still move the needle significantly. It's certainly how we've gone in. We were actually, as we did our trial, dosed up to 8 mg per kg, at which point we're at a 64-fold molar excess of the approved dose of Proleukin with a half-life going out days, seeing NK activation, seeing tumor-specific T-cell activation. So we're really excited about some of these metrics that are emerging. And I would say we, as a field and as a sort of a continuum of this, have got to just continue to build on this, like others have said here.
Yeah, and maybe sorry, Dr. Diab, go ahead.
Yeah, so I'll be quick. There's 3 points I want to make about the comments from Navim. So in terms of what happens to the field if it's negative or positive at trial, from an investigator point of view, we hope that things don't follow the IDO pathway and go to the skeptic mode, but we know the investor world is very volatile, and we're worried that if there is negative at trial, that's going to happen or that's going to impact the field. We hope it's not, because these technologies that are just presenting to us right now and out there are very exciting to the investigators, and I think, yes, this more companies makes the field better. So hopefully not. I think positive trials, everybody agrees, is going to ignite the field even more and more. So let's hope that will be the case. The second point about increasing single agents activity without the toxicity, we have to be careful about the negative feedback, and that's point you mentioned, that activation-induced cell death is one of the mechanisms that restrain IL-2. So we have to be careful, although some people think that's mediated through the alpha subunit of the receptor. Some people don't, because we see it in somewhere else. So we have to be careful of where is that optimum of the single agent super activation, even without toxicity. And the third point is just from advice of drug development point of view from the investigator side is just look at the lag, and this is about the design of the trial. Single agents activity is always 60. Nobody argued with the 6 appeal of single agents activity, but where you can get it is very important. The LAG-3 trials that shocked the field and its positivity right now. If you look at the LAG-3, it was developed initially in the refractory melanoma with a response rate of 13% that did not excite most of people. On the other hand, the phase 3 trial on the treatment naive, we know its positivity with combination. And the LAG-3, the BMS one, did not have a trial of single agent activity that I know about, but there are some trials, I think Novartis has and others, that did not show any single agent activity by itself. So that tells you that the combination can still happen without single agent activity. But the design of the trial, in ASCO 2021, there was the presentation of the phase 3 that was positive LAG-3 plus Nivo versus Nivo. On the other hand, the one that mimicked that result and was close to the result is the development of LAG-3 plus Nivo in the neoadjuvant setting was very impressive data compared to the LAG-3 plus Nivo, the same agent, in the refractory setting. So while a lot of people going for that high bar of refractory setting, which is really sexy to achieve it, it might not be the right way to develop your drug. So that's a real world example of the LAG-3.
Yeah, understood. Yeah, there are two, conflicting factors there. One is the cost of doing studies in earlier stage patients, and two is the regulatory. environment. But I think most people, I see most people nodding their heads in agreement on the screen here. So I think there's a consensus there. We've only got a couple more minutes left, and this has been dominated by our discussions about IL-2. But maybe we could ask. I'd like to get some of the commercial guys to comment. Jennifer Cullinan's working with IL-12. John, you're working with 2, 12, 15. Randy, you guys are 2 and 12. Fong, I'm sorry, I forget if you're working beyond IL-2, but maybe just tell us about how you rank order. Sort of, it seems to that IL-2 is kind of the alpha dog. Sort of like PD-1 is for checkpoints. IL-2 is kind of the alpha dog, but we have a decent hierarchy of other intriguing cytokines. So, how did you guys think about what else to develop beyond IL-2?
Yeah, Mike. I mean, I think of it as.
Let me let John go first.
I think of it as each one is its own hypothesis, right? So there's been a tremendous amount of work on IL-2 because of the Proleukin had it had single agent activity. The Nectar data that was disclosed a couple of years ago, the phase 1 data got people excited. But one thing to be very clear about IL-15, depending on how you make it, right? The way we've built it, it's an IL-15 receptor alpha heterocomplex. That's going to behave almost exactly like what people call a not alpha IL-2, right? It interacts with precisely the same receptors. IL-12 that's one that people want to tame the beast, right? It's like an extraordinarily hot molecule. It's going to be less proliferative than IL-2, but it makes T cells and NK cells highly cytotoxic. And so the question is, can a company like Xencor or now BMS with Dragonfly, can you tame it to the extent that you can actually explore those activities? And that the super strong interferon gamma response has all kinds of potential to light up the tumor microenvironment. Yes, it's going to upregulate PD-L1, but I think we know how to solve that problem. But it's going to upregulate class 1, and and it has a lot of potential to completely alter the TME.
I would totally agree. I mean, IL-2 is really good on the lymphoid axis of the immune system, but IL-12, it's a crossover. It's very early in the immune cascade. It'll activate, stimulate myeloid cells, and they'll secrete more IL-12, and it has this whole feed-forward mechanism.
Yeah. It's almost like IL-2 is the booster, is the of the rocket, and then IL-12 is the second stage that could take it to the next.
And don't forget IL-18. That one's also interesting.
Yeah.
Michael, I'd like to make
Go ahead, Erin. Yeah.
Thank you. So Werewolf also has an IL-12 indokine molecule, which comes out of the Predator platform, masked, systemically delivered, masked in the periphery, tumor microenvironment delivered, fully potent IL-12. And we've seen some tremendous activity in preclinical models. There are non-overlapping immune mechanisms, as you know, for the various cytokines. IL-12 affecting both the innate and adaptive immune system has the potential to initiate an immune response and not just amplify one.
Right.
Potentially finding clinical utility in tumor types that are not historically sensitive to immunotherapy agents. So I think, I see IL-2 as sort of the starting point, but I see IL-12 a little bit like the Holy Grail, and expanding even beyond, especially if we can harness it to avoid the systemic toxicity.
And how important is its activity on NK cells?
I think it's very important. I think it's as important as its activity on T cells.
It's funny, Randy. I always use the term the Holy Grail for IL-12.
It is the Holy Grail.
Yeah, maybe if I could jump in and just tell a little bit about Cullen Oncology. We're actually really excited because we have a single agent therapeutic that combines both IL-2 and IL-12, and we really see, it's clear from the literature as well as our own work that there's real synergy actually between IL-2 and IL-12, and the combination really has manifested itself in terms of the ability to, in both cases, stimulate both a wide variety of T cell subsets as well as NK cells. And there's synergy in terms of, for example, activation of IL-2 upregulating the IL-12 receptor. There's orthogonal induction of the JAK-STAT pathways that really synergizes as well as induction of complementary cytokines, antitumoral cytokines like interferon gamma, TNF, et cetera. And you can see, and certainly the evidence from the in vivo models has shown the synergy as well. And we actually use very challenging preclinical models like the B16F10 model that typically for, a single cytokine, you can't really see single agent activity, but with our IL-2, IL-12 constructs delivered intratumorally with the tumor retention, we see really dramatic antitumoral responses, including complete responses in very challenging models. So that's sort of our lead molecule, CLN-617. But, I agree that there are other exciting potential for combinations as well with the other combinations, for example, the IL-18, et cetera, that could also be built into our platform for delivery of two cytokines in one.
Yeah, one other thing that we are working on is blockade of IL-4/13. And this is basically, we feel, is another space that's been not looked at. I think there's plenty of data with Dupixent, dupilumab, which is already approved for atopic dermatitis. And recent transactions in the CD47 space where you're trying to repolarize or change the polarization of the macrophages to the M1 phenotype and make it more inflamed. We feel that we've sort of initially demonstrated to be very beneficial. We've also shown with IL-4 targeting with our glioblastoma project clearly has been able to show really impressive survival outcomes in these patients. So I think one other space, obviously quite an orthogonal approach here, where in addition to having the ability of IL-2 to stimulate your cancer-fighting T cells, the ability to, at the same time, to create an inflamed tumor microenvironment by preventing proliferation of MDSCs, as well as having the M2A macrophages be driven towards the M1 phenotype. That, I think, is a really good 1-2 punch. We think will be an exciting space to watch out for as well.
Will Redmond, I know you've published on the IL-4 space. Can you add to anything that Bahar had mentioned?
Yeah, no, I think it's a great point. And I would add, our work in that space was looking at combinations of OX40 agonists and checkpoint blockade and seeing that actually induced a Th2 IL-4 response. And if you block that, you further improve outcomes. So I think I would just add that we know, and we've published, that IL-2 receptor and OX40 receptor signaling co-regulate each other. So I think there's a lot of opportunities if you're giving IL-2 agonists. It's going to drive other co-stim molecules. Things like IL-12 also are cross-regulated and by those pathways. So I think there's a lot of other potential combinatorial options beyond just the cytokines or combinations of cytokines that could really, certainly preclinically, you get tremendous responses, and to see that hopefully moving forward into patients would be wonderful.
Okay. Unfortunately, I have to wrap it up there, guys, because I'm going to turn into a pumpkin any second. But I really appreciate it. I've just got to do my little closing spiel and just thank everybody for participating. This was everything I hoped it to be and more. Appreciate everybody's time and enthusiasm that everybody exhibited to contribute to this. And this could be a whole day's worth of, which gives me an idea, of discussions. But thank you, everybody who was watching online in our investor audience. Hopefully next year we'll be able to do this in person rather than virtually. We thank you again from both myself and the H.C. Wainwright team, and wish you a good rest of day. To everybody participating on the panel, let me once again thank you for your time, and please have a good day.
Thanks, Michael.
Thank you, Michael.
Thank you, Michael. Bye.